Hello student, welcome to EPG Pathsala. I am Dr. Ajit Jaswal from Department of Anthropology, Pondicherry Central University, Puducherry. Today we are going to discuss about a module called as Milestone in the field of genetics, under paper called as Human Population Genetics. The main objective of this particular modules are as follows. In this modules, we are going to discuss about the important milestone in the field of molecular genetics and also major milestone in the field, specifically in the case of genetics. We'll try to study the historical concept of gene therapy and also try to highlight the features which revolving around the principal concept as well as development of genetics, like genome wide association studies, resource to keep up with the field, and so on. Okay, now before going to discuss in detail regarding the milestone, let me give you the introductory or initial information about this. When we search the published literature, we can see an exponential increase of literature on genetics in recent decade, which makes it one of the world's leading sciences. Most of the popular sciences in the media seems superficially to be about other subjects, but genetics is the indispensable platform on which almost all life and health is based. Although it is quite difficult to say which discovery is most important from the beginning of this subject. The actual birth of genetics occurred around 60 years back when Watson and Crick published Structure of DNA. But a few of them stand out in the way that the illumination of the genetic code or the transcriptional mechanism of the lac operon did in the late 1950s and early 1960s, whereas genetic code or triplet was published in 1961. In 1977, it became clear that eukaryotic genes are interrupted due to the presence of exons and introns. It is clarified that various portions of DNA do not show transcriptions and translation. In the same time, the DNA can be sequenced. PCR or polymerase chain reaction, a technique developed by Carl Mullis in 1983, is now a common or often indispensable for medical as well as and biological research. DNA cloning for sequencing or DNA-based phylogeny or functional analysis of gene hereditary disease. The identification of gene fingerprint, which is used in forensic science and paternity testings, and the detection and diagnosis of infectious disease became easy by the development of this technique. In 1993, Mullis awarded the Nobel Prize in Chemistry along with Michael Smith for his work on PCR. When we discuss about the milestone in molecular genetics, like starting from 1928 or even before also, but in 1928, Fred Griffith provides evidence for genetic transformation factor like DNA. Similarly, later on in 1944, Avery's work, Avery, MacLeod and McCarthy, they determined DNA is the genetic material. In 1952, Hershey and Chase also determined that DNA is the genetic material. But the important work of Watson and Crick in 1953, they explained the structure of DNA. However, the important contribution like in 1970 provided the first restriction endonuclease isolated. So in 1970, first restriction endonuclease was isolated. However, in 1973, Boyds and Cohen established recombinant DNA technology, and this technology was this particular study is further developed in 1976 in the form of DNA sequencing technique, In 1980, US Supreme Court rules that genetically modified microorganisms can be patented. If you see the whole story once again, in 1976, Walter Fires was the first to establish the complete nucleotide sequence of viral RNA, completed the first pair only. In the mid of life were released. New sequencing technologies such as massive parallel sequencing technique emerged as very easy over sequencing in the last decade. In 2001, Genome-Wide Association Studies (GWAS) has documented a strong statistical association between common genetic variation at thousands of loci and more than 250 human traits. The functional effect of most GWAS implicated variants are not explained till this date. The finding that nearly 90% of these sites occur outside of protein coding sequence suggests that associated variant may instead have a role in gene regulation. Genetic variation can also, the further studies explain that genetic variation can also influence gene expression through alteration in splicing, non-coding RNA expression and RNA stability. Analyzing multiple tissue will be important because evaluation of the functional consequences of a disease-oriented SNP is ideally performed in a disease-relevant cell context. The technique for genome mapping and sequencing. Again, if we look toward the earlier part of information for most tissue types, human biospecimens are very difficult to obtain from living donor. For example, from brain, heart and pancreas, and most EQTL studies so far have been performed with RNA isolated from immortalized lymphoblastic or lymphocysts and a few additional readily sampled tissue. So, from the technique of genome mapping and sequencing, we can see the gradual increase or the changes of the biotech industry all over the world is clearly are driven largely by the discoveries from molecular biology. The expression for betterment of people need have not been fulfilled. We can say clearly that such scientific development, such genetical upliftments, such biological or biotechnological discoveries play a very active role towards the applied aspect of human improvement or human betterment. This technique provides are helpful for eradicating or removing, curing, correcting some of the disease or disorders or problems in a much more simplified or better way in comparison to the earlier technique where such informations were altogether missing. When we discuss about the major milestone in genetics or milestone in molecular genetics, as we all know that the first DNA was synthesized sold in 1981. The PCR method was published in 1988. The further historical information development like Human Genome Project was initiated in 1990 and in 1996 complete DNA sequence of a eukaryote yeast were determined. In 1997, nuclear cloning of a mammal, that is a sheep named Dolly, was completed. In 2000, Arabidopsis genome sequencing was done. 2001 was a great year for the molecular genetist because during this year human genome sequence was performed. In 2002, complete human gene microarray, what we call as gene chips, were available. Major milestone in genetics. Here are some of the important features that we have to understand before moving toward the historical aspect. Like genomic is a complete reading of DNA sequence. Next, silencing gene is the new genetics. Next, biomarkers are the discovery of extreme habitats. Next important point, synthetic biology include biobricks. Then building novel biological systems from interchangeable and standardized parts. And another one important point is a registry of standardized DNA biobricks, because biobricks can be assembled to form devices and systems to operate in living cell. So the major milestone, if you see from the historical point from 1855 to till today, the important one is as follows. In 1855, E. coli, called as Escherichia coli bacterium, was discovered, later becomes a major tool for biotechnology. In 1973, Cohen and Boer, they developed a genetic engineering technique to cut and paste DNA and reproduce the new DNA in bacteria. In 1975, Kohler and Milstein, they developed hybridoma technology and they got the Nobel Prize in 1984. Similarly, in 1977, Genentech scientists and their collaborators produced the first human protein called somatostain in a bacterium called E. coli. Further research in 1982 by Eli Lilly and Company, they market recombinant human insulin, known to be the first such product on the market. The first therapeutic monoclonal antibody approved by FDA, Muromonab, for organ transplant rejection prevention, was developed in 1986. 2009 is known to be a major milestone in the genetics as first tissue engineering product approved in the EU, European Union, chondroclic. Similarly, the first gene therapy approved in the EU called Glybera in 2012. Now let's discuss about the history of gene therapy or historical changes related to gene therapy. In the year 1977, a gene was successfully delivered into mammalian cell, and similarly in 1990s, first human gene therapy was approved, called as by SCID. Similarly, after 9 years, that is in 1993, Gelsinger with OTC deficiency died from a organ failure after gene therapy. In 2000, A. Fisher, he cured children with SCID using retroviral vector 2 of the children. 6 years after this this particular disease or retroviral 2 of the children developed leukemia. FDA halted the use of retrovirus in the US. 6 years after, that is in 2006, patients was successfully treated with metastatic melanoma using killer T cell genetically retargeted to attack the cancer cell. Similarly, in the same year, there is a successful gene-based TH for the treatment of HIV, lentiviral vector for delivery of an antisense gene against HIV developed. After few years, that is in 2009, researchers succeeded at handling adenoleukodystrophy using a vector derived from HIV to deliver the gene for the missing enzyme. So the history of gene therapy clearly shows the historical changes or revolutionary changes took place from 1977 till today, even the recent one is till 2009. Molecular therapies. DNA modulated therapies are like gene replacement, gene transfer, gene editing, gene shifting. Gene replacement related to replace the defected gene. Gene transfer is related to upregulate therapeutic protein, whereas gene editing is to correct the defected gene. When we talk about gene shifting, it is related to upregulate healthy mitochondrial DNA molecules, change ratio of heteroplasmy. Similarly, when we discuss about R and RNA modulated therapies, it may be exon skipping by antisense oligonucleotides or exon inclusion by antisense oligonucleotide, maybe because of mutant RNA removal. It is also because of inactivation of the mutant mRNA by RNAi. Similarly, RNA modulating therapies also related to destruction of the mutant mRNA by ribozymes. Overall, so if we try to once again recap the whole milestone in genetics, let's discuss about protein modulated theories. In this, the important concept is related to neutralization of a mutant protein, regulating the level of haploinsufficient protein, and upregulation compensatory molecules. So, protein modulating theories revolve around these three concepts: neutralization, regulations, and upregulations compensation molecules. When we discuss about genetics as an information science, when we see the historical milestone, the molecules of inheritance is DNA and not protein. Remember this one. This was confirmed in 1944. In 1944, it was very clear the molecules of inheritance is DNA, not protein. This information was further strengthened by the work developed by Watson and Crick double helical model. The Watson and Crick double helix model of DNA, it permits transcriptions and replication along with mutation. This information came in 1953. In 1956, immediately after the Watson and Crick model, they were very clear that there is 46 chromosome, not 48 chromosome in human being. The triplet code for protein demonstrated in 1960s. The principle of unity in diversity applies to all living things at all levels, from molecules to cell to organ function to ecosystem. System biology combines the digital code of genetics with environmental and also with behavioral inputs and perturbations. This information was given by Leroy Hood. Latest modern, the synthetic biology, which is given by George Crunch, that also developed a new phase in the life. Similarly, the stem cell, the development of stem cell, which framed a revolutionary improvement in the field of molecular genetics or genetic engineering or molecular science. Stem cell global genomic analysis leads to the definition of stemness, that is the assemble of the genes that are necessary and sufficient to convert any cell, even the skin cell, into a stem cell. From this, it is very convenient. From this converted stem cell is possible to obtain living plant and animal also. We can explain in a somewhat better way with the concept like the changes that are the evolutionary transformation that took place as in 1972 the first recombinant DNA discovered. Scientists describe restriction nuclease enzyme that recognize and cut specific short sequence of DNA. The resulting fragment can be used to analyze DNA. After these enzymes later become an important tool for mapping genomes. Scientists produce recombinant DNA molecular by joining DNA from different species and subsequently inserting the hybrid DNA into a source cell, often a bacterium. In 1972, Burks constructed the first recombinant DNA molecules in vitro. Boyers and Cohen's first cloning of a recombinant DNA molecules. 1973 discovery, the first animal gene clone. Researchers fuse a segment of DNA containing a gene from the Africa clawed frog Xenopus with DNA from the bacterium E. coli and place the resulting DNA back into an E. coli cell. There the dog DNA was copied with the gene it contained directed the production of a specific frog protein. Invention by Mullis in 1986 of the polymerase chain reaction to amplify specific DNA sequence. Completion of genomic sequence for an increasing number of organisms has spawned the new field of genomics. Knowledge of individual genes and their regulation will be important to basic biological research as well as to specific applications such as medical genetics. Powerful new technique in genetics raises important ethical, legal and social issues that will need thoughtful solution. In 1975, DNA sequencing discovered two groups, Frederick Sanger and colleague and Alan Maxmund Walter Gilbert both developed a rapid DNA sequencing method. The Sanger method is most commonly employed in the lab today, with colored dyes used to identify each of the four nucleic acids that make up DNA. First gene engineering company discovered in 1973 by Herbert Boyer founds Genentech. Gene Bank database formed in 1982, scientists began submitting DNA sequencing data to a National Institute of Health database that is open to the public. First disease gene mapped in 1983. 1990, which is known to be the launch of Human Genome Project, the Department of Energy and the National Institute of Health announced a plan for a 15-year project to sequence the human genome. This will eventually result in sequencing all 3.2 billion letters of human genome. EST's fragment of gene discovered in 1991. Expression tag called EST, an identified piece of a gene is made by copying a portion of mRNA or messenger RNA molecule. As such, EST provides a way to focus on the expressed portion of the genome, which is less than 1/10. In 1992, second generation genetic map of human genome discovered. A French team built a low-resolution microsatellite genetic map of the entire human genome. Each generation of the map helped geneticists more quickly locate disease gene on chromosome. Flavor Saver tomato discovered in 1994. The Food and Drug Administration approved the sale of first genetically modified food. Tuberculosis and tuberculosis bacterium sequenced in 1998. Mycobacterium tuberculosis causes the chronic infection disease tuberculosis. The sequencing of this bacterium is expected to help scientists develop new therapies to treat the disease. In 1998, a roundworm C. elegans sequenced the first genome sequence of a multicellular organism. The roundworm is completed. Chromosome 22 sequenced in 1999. The first finished full-length sequence of a human chromosome is produced. Chromosome 22 was chosen to be the first because it is relatively small and had a highly detailed map already available. Such a map is necessary for the clone by the clone sequencing approach. Personalized medicine. When we talk about the personalized medicine, here we discuss about drugs that work all the time. There are not much drugs are available that work all the time. We also discuss about personalized medicine. There is a majority of the drugs that work sometime, that occasionally, or when the condition prevails, or at very limited specific period. There are some of the medicines that won't work, called as frauds. So the medicines, personalized medicine, may be there are very few in number who work all the time. Majority of the medicines, they work sometimes, that specific conditions, and there is some medicine which is called as frauds, as they don't work. So the drugs that work sometime, that is in the case of majority, individual differences, there is a sometime individual differences in the genome occur. They are somewhat such drugs are found in more than 1 lakh points. These differences defined as a risk of disease as well as response to drug. So the concept of risk of disease and response of drugs is also an important concept in personalized medicine. Rational genetics. Rational genetic analyze the result of crosses by looking directly at the genes, like the in-not genome and the white Vidya-mitra. So the rational genetics is concerned about analyzing the result of crosses mainly by looking directly at the gene, not any other way. Mapping the human genome sequencing. The human genome started in 1990 and continued till 2000. Another started in 1998 and it continued till 2003. Interpreting the human genome sequencing up to 2003 to maybe continue. The Human Genome Project is generally beyond the, either it may be beyond the Human Genome Project. There is some explanations on that. Actually, it is arranged on multiple chromosome, or what we call a human genome is arranged on multiple chromosome, which consist of 23 pair of chromosome, in which 22 pair is known to be autosome and 1 pair called sex chromosome. As in the case of female, it may be XX or it may be XY in the case of male. As we all know that humans have 23 pair of chromosome in every cell except matured RBC. Gametes are sex cell, which is called as sperm and egg, have half the normal complement of chromosome. Human Genome Project is an international project aiming for sequencing and localizing of the base sequence that make up human DNA. Human Genome Project (HGP) is an international project aiming for sequencing and localization of the base sequence that makes up human DNA. Store this information in database. So mapping of human genome require a set of landmarks. Some of these landmarks are gene, but many more are nameless stretches of DNA, such as RFLP, VNTR, and STS. In 1990s, American geneticists started an ambitious quest to map and sequence the entire human genome. 9 years after this, in 1999, the first draft of human chromosome 22 developed. 2000, the final draft of human chromosome 21 developed. In 2000 working draft of the whole human genome. Similarly, in 2004, the finished sequence of the eukaryotic part of human genome. So, if you see the whole process, 1999, American geneticists started an ambitious quest to map and sequence the entire human genome. In 1999, final draft of human chromosome 22 developed. 2000, final draft of human chromosome 21 developed. 2001, working draft of whole human genome, and ultimately in 2004, the finished sequence of the euchromating part of the human genome completed. Genome-wide association studies GWA. Genome-wide association or GWA studies represent a systematic search with nucleic acid probe called chips for variant in the genome statistically associated with particular disease or traits. We can also explain it as a next generation sequencing is replacing chips array. Only 2% of the DNA codes for protein product. So, few of these variants actually occur in such coding genes, but they may still influence regulation of gene function. Tremendous investment and output past several years has transformed the genetic side of molecular epidemiology, but neglected non-genetic variables. Variant gives clue to unsuspected genes and pathways potentially involved in disease like diabetic mellitus. Resources to keep up with field. US NIH, what is called as NCI NHGR, maintain an ongoing catalog of published genome-wide association studies. There are many databases of gene sequencing and variant and the protein variant to assist in annotations of the potential biological role of variant in or near mapping gene. Statistical compendia for test and adjustment for bias due to selection, misclassification and population stratification are established. GWAS graphical user interface, graphical browser are known to be the resources to keep up with field. Avalanche of genomic information. The International HapMap Consortium aims to genotype 1 million SNP from 270 individuals. Direct association of individual with SNP allele with disease phenotype including linkage disequilibrium LD are more powerful than linkage based indirect association analysis. DB-SNP has more than 10 billion validated SNPs. Haplotype structure can be obtained via genome-wide LD, haplotype block 1 kb to 1 MB, and haplotype tagging SNPs, respecting recombination hotspot and variable LD. Now let's discuss about estimated cost of genotype. When human genome sequence published in 2001 along with 10 million common SNPs identified, proposed case control studies of 100 plus 100 participants with 20 billion genotype at the rate of about 0.50 had cost estimated of about $10 billion. HapMap brought cost of 3 lakh tagging SNPs at the cost of about 0.003 to $2 million per common disease. That is, if you estimate 500x decrease in 4 years. Now we have even more powerful analysis with next generation sequencing of the genome. Computational muscle. Skate where the puck is gonna be in planning big studies. A golden age for public health science. Sequencing and analyzing the human genome is generating genetic information that must be linked with information about nutrition and metabolism, lifestyle behavior, disease and medications, microbial, chemical, physical exposure, and every discipline of public health science needed. Now let me give you the summary of the whole section. It is likely that genetic engineering will remain unpopular and controversial so long as it remains a centralized activity in the hand of large corporates. Even the domestication of biotechnology will dominate our lives during the next 50 years. We also find out that we have seen that genetically modified tropical fish with new and brilliant colors appeared in pet stores, similar to the world of natural breeders. The next step is to become user friendly. So, in the era of open source biology, the magic of genes will be available to anyone with the skill and imagination to use it. Thank you.